In this video, we'll talk about statistical analyses and how they are conducted in fMRI. We'll start with a t-test, which gives us a very simple way of conducting analyses and block designs. So the ultimate goal is to obtain these kinds of maps where each voxel is represented by this overlay here and shows in the colored voxels those voxels that survive some statistical threshold. So an alpha value here, right? So in this case, let's say this is a block design that's generating this map where we look at the difference between a control and a treatment condition. And those voxels are showing significantly greater signal in the treatment condition compared to the control condition. So the goal here is, what's the observed difference between conditions, and is this difference reliable? And for this, we just need standard statistics that are applied to fMRI data, and they're applied on a voxel by voxel basis here. This is obviously not ideal, but we can correct for the multiple comparisons that we're conducting. So the significance testing is then computed and a color scale and on a voxel by voxel level. And this color scale is used for each voxel to visualize whether this voxel surpassed some statistical threshold. The threshold is called an alpha value, and voxels are labeled as significant, so they're colored in with different colors representing the level of the p-value or the magnitude of the p-value. And voxels above the threshold are labeled as non-significant, so they're not colored. So only voxels that are below the threshold of a specific p-value, in this case is 0.001. This used to be the standard in older papers and still sometimes can be used as a standard, but it's not ideal. We want to correct an alpha value for multiple comparisons. So the alpha value that we adapt needs to correct for the 200,000 comparisons that we make. The problem here being that the more comparisons you make, the more likely it is that a number of voxels simply survive the threshold or are below this pre-specified alpha value due to chance, right? There's always some level of chance that would lead to us stating that a voxel is significantly activated, whereas in actual life it is not. So this is what's indicated here by this 2x2 matrix, with the hypothesis truth at the top and on the left side output of the statistical test. So we can have 4 different cases. The truth being that the alternative hypothesis is true and we reject the null hypothesis, right? That would give us a hit. But if we reject the null hypothesis and the null hypothesis is true, we get a type 1 error, or a false alarm, false positive, which is something we want to avoid. And that's what we control in our alpha value in our test. It can also be that the hypothesis is true. So we should say that voxels are active, but we actually accept the null hypothesis. This would be called a type 2 error. And this is related to the power of the experiment, with higher power minimizing type 2 errors. Alternatively, the null hypothesis might be true, and we accept the null hypothesis. This is a correct rejection. So we want hits and correct rejections, obviously, and we want to maximize those. This means that our significance and non-significance statements are associated with errors, right? Type 1 and type 2 errors. A type 1 error is the probability of deciding that the null hypothesis is false when it is really true. So this would be a false positive. The voxel is colored, but it should not have been colored, right? So we're saying that the voxel surpasses or is below the threshold, below the alpha value, but it really shouldn't be. And this is something we increase with increasing comparisons that we conduct. And per brain, that's about 150 to 200,000. The type 2 error is the probability of accepting the null hypothesis when it really is false. So that's a false negative. It's when we don't color the voxel when it should have been colored. This is also something that's unfortunate for the researchers, because then they make wrong conclusions about something being absent when it really shouldn't be. So we need to make compromises when it comes to multiple comparisons correction. And we will discuss this. There are some methods now that allow us to control for this. such as random Gaussian field theory. But let's talk about the t-test for a second here. So let's consider a simple experiment in which visual stimuli were shown to the participants at specific time points. And we want to make a decision about whether at these time points, immediately after the stimulus was presented, is the signal significantly larger than at null time points, so when nothing was presented to participants. So we can see this here now, and this is a sort of stylized time series, which we almost never see in experiments. So we could see here just by eyeballing this that this is significant, but real experiments are much more complex than this. So a simple way to perform this statistical test in a more complex experiment, where we cannot eyeball whether a treatment condition is different from a control condition, is to simply perform a t-test and to test the null hypothesis in the case when there are only two conditions. The null hypothesis, or H0, is basically saying that the activation during treatment conditions is equal to the activation during control conditions. And this would mean that there is no condition effect on fMRI responses. In this case, we can simply create a t-statistic for the fMRI data. Well, we know that the hemodynamic response is very slow, so how can this be done? What time points should be assigned to each condition in this type of experiment? And here's an example of a simplified version of this. So we know when we present the baseline conditions or the control conditions, and we know the time points at which we present the task conditions or the treatment conditions. And this is showing this basically in red. Red is baseline, blue is task, red is baseline, blue is task. Then we can compute the mean for task conditions, and we can compute the mean bolt response for baseline conditions. And in a t-test, we would simply subtract the mean of one condition, so let's say task minus baseline, and divide this by the pooled standard deviation. The pooled standard deviation in this t-test is simply the variance, so the sum of the squared deviations of each score from each mean, weighted by the number of observations per condition. So this is sigma xy here, right? In this case, we have a task response of 6.3, a no task response or baseline response of 4.3, and a pooled standard deviation of 0.87, giving us a t-score of 2.3. So given that this is such a large pooled standard deviation here, we have a relatively small effect size here, which may not be significant depending on the number of observations that we have. We have another example here in which we have a baseline task that has a lower response. You can see that the difference between these two is a little bit lower, but we have a very small standard deviation term here. So that leads to a much larger response given that the pooled standard deviation is so small. So you can see that that in a t-test we take into account the difference between two conditions in the signal and the average signal between the two conditions, but also the standard deviation across both types of time periods, if you will. So task and non-task time points lead to a pooled standard deviation, which is then the denominator in the t-test. That is, in other words, we can say, and we had discussed this earlier, that we're interested in between conditions variability. Remember when we talked about functional signal to noise ratio, which is the variability between a baseline condition or a control condition and a treatment condition. So that is something we'd like to maximize if we want significant results. But we also have to deal with these with the within condition variability that's shown here. So the variability while the task is on and the variability in the signal while the task is off. So just within a given condition, the variability determines within conditions variability. And the noise then basically is computed as the pooled standard deviation based on this within conditions variability, whereas the signal would be the between conditions variability. That's exactly what we also just discussed when we talked about the t-test. So as we reduce the noise, the within conditions variability, we're increasing the t-statistic basically. Also, as we increase the difference between task here and baseline here, we also increasing the t-statistic and the possibility to detect a significant effect. So one problem when it comes to considering t-tests and block designs is which time points to use. So we know when a condition started and when a condition ended. So let's say these are task conditions and these are non-task conditions. So we need to decide which time points to use here that we want to analyze. So where do we get the signal from and where do we get the variability from also? And one strategy is to use a lag. So we don't want to use immediately the onset because we know that it takes some time for the hemodynamic response to reach its amplitude, and then also it takes some time to go back down to baseline. So we want to exclude those transition points usually and use only the middle portions of each block. And this is basically what's illustrated in this step. So while you might initially want to just start analyzing the data from the beginning as soon as the block started, this is probably not a good strategy. You want to shift this by about 6 seconds, so as long as it takes to reach this plateau here. And then you want to cut the transition points off also, so that you're only analyzing signal from the parts that are actually activated based on what we know about the lag of the hemodynamic response. So in the red, we have the treatment mean and standard deviation that we extract from only those time points. And in the blue, we have the control mean and standard deviations that we'll extract only from those points. We'll ignore the periods when the signal rises and the periods when the signal falls. So we're only analyzing red, blue, right? This helps us make sure that we're only considering signal that is actually meaningful by considering the lag that it takes and excluding the transition points in the middle of these types of block designs. So between block 1 and block 2. The next question we can ask is, well, can a t-test be used to analyze event-related data? And it really depends on what type of event-related data we're talking about. Typically, we want to use a general linear model, which we'll talk about in another video, because they're relatively complex. They include multiple time points, but it is also possible to use a t-test to analyze event-related data. In this case, what we can do then is to first perform signal averaging for different types of trials. In this case, which is a study by Luis Pessoa that was published in Neuron in 2002, we're considering correct trials and we're considering incorrect trials. The correct trials are shown in red and the incorrect trials are shown in blue. And the region from which we're extracting signal is the right frontal eye field. And then we can again consider a specific period from which we extract mean and standard deviations. So what we'll do is we average all the incorrect trials, we average all the correct trials, we get an average response for this, and we also get a standard deviation for each of these responses. And then we can again use a t-test to look exactly at this, at the difference between those 2 time points here and those 2 time points here. So we average also across these 2 time points. And the t-test allows us to indicate or identify whether the red line is significantly larger, the response relative to the response represented here by the blue line. So t-tests can be done, but averaging first helps to extract the hemodynamic response for 2 conditions.